And then I'll be very happy to have Dr. Winn present. Dr. Virginia Winn is the lead for our basic science discovery domain. She's also the director of reproductive stem cell and perinatal biology in the OBGYN department. She's a physician scientist and a fantastic investigator who has a passion to investigate the human placental development, and that's the focus of her lab.
Great, thank you, Anna. Happy to be here, excited to share some of our work with you. What I thought I would do is start with just a basic overview of 3 of the larger domains within my research group of the areas that we investigate. We are truly a basic and translational research program. On the basic side, we spend a lot of time trying to understand human placental development, specifically trophoblast invasion and vascular remodeling, particularly as it relates to preeclampsia. We're also very interested in understanding communication of the placenta to the maternal system through understanding more about extracellular vesicles or exosomes, as well as utilizing maternal blood as a window into assessing placental health and function over pregnancy by measurements of not only exosomes, but cell-free DNA, cell-free RNA. And lastly, very interested in how potentially these extracellular vesicles or other factors are impacting the maternal immune and endothelial systems in pregnancy, and specifically related to preeclampsia pathogenesis. And so I will actually just going to share with you one of the projects that we have ongoing related to this 3rd domain of understanding endothelial dysfunction related to preeclampsia. So just a quick background, hypertensive disorders of pregnancy impact 8 million mother-infant pairs worldwide and accounts for 76,000 maternal deaths and probably about 500,000 fetal neonatal deaths worldwide. And so this is just a distribution of the incidence of HDP across the world. In terms of the pathophysiology, it's usually thought about occurring in 2 stages. Preeclampsia is characterized by new onset hypertension and proteinuria or other end organ damage that occurs in the 2nd half of pregnancy. But we think the pathogenesis begins earlier than that in the 1st half of pregnancy, and often can be characterized by abnormal placentation that then results in the maternal presentation of preeclampsia. And what we're going to be focusing on today is understanding this endothelial dysfunction, which is a key central part of the maternal presentation of preeclampsia. And one of the pathways that we're interested in looking at related to preeclampsia pathogenesis is the hemoxygenase pathway. Hemoxygenase is a enzyme that converts heme into carbon monoxide, iron, and biliverdin, which very quickly gets converted to bilirubin. This is actually an important pathway, has been shown to be involved in endothelial function, hypertension, cardiovascular disease, and also potential role in preeclampsia. So the hemoxygenase is expressed by the endothelial cells, which line our blood vessels. And its activity is important in decreasing inflammation, protecting against apoptosis, and enhancing proliferation. So what do we know about hemoxygenase 1 in pregnancy? There's a mouse model system of partial deficiency in hemoxygenase 1 activity that results in impacting blood pressure, and also has fetal IUGR and placental vascular changes that are reminiscent of human preeclampsia. It's also been shown that reduced placental HO1 levels are associated with preeclampsia, spontaneous abortions, and recurrent miscarriage. And interestingly, preeclamptic women have decreased end-tidal carbon monoxide levels, which is that end product of hemoxygenase activity compared to normotensive women. So what do we know about the hemoxygenase 1 gene? It actually turns out that there are two key regulatory human alleles that occur upstream of the start codon in one transcript and an intron, but also has regulatory components. And this SNP located here at -413 is a known human variant, as well as there's this GT repeat region that can be very different sizes. And it's been demonstrated that these alleles impact gene expression levels and therefore hemoxygenase 1 activity, with the A SNP linked to higher levels of activity, and shorter GT repeats linked with higher activities, which we would consider protective for the cells. So we were interested in looking at this gene, and one of the first questions was, what is the racial and ethnic distribution of HO1? And we turned to the 1000 Genome Project, which is a publicly available genome sequencing data set. Unfortunately, it doesn't have a lot of clinical data, but this is to give a good idea of distribution of genetic variants across the world. From this population, we downloaded the female genomes, and they are represented different regions of the world. Yes. African is represented both by US African population and African populations. The Hispanic is represented by the Americas. Europe is Caucasian, and then we have the South Asian and Eastern Asian populations. So Tien Sun, a postdoc in my lab, in collaboration with a lab at UCSD, was able to, from this data, determine the number of GT repeats using the Gremic lab pipeline. And then also the AT SNP is easily determined from the ensemble. And then as we get further into the talk, we were able to get a hold of a hypertensive disease and pregnancy cohort from the Preeclampsia Foundation, and Tien actually sequenced and determined the GT repeats and the AT SNP of those genes. So if we look at the 1,000 Genome Project, the number of GT repeats in that whole population is graphed here as frequency in a histogram. And you can see that there are ranges from 18 up to 42 repeats, but clearly there's certain repeats that are much more frequent in the populations. And then in terms of the AT-SNP distribution in the 1,000 Genome Project, you can see the ratios and frequency ratios here. Looking at, these are the numbers that we had data from each of these. So when you look at the GT repeat by the racial groups as defined in the 1,000 Genome Project, you can see that their histograms look different across the different racial groups. Particularly the African population has much higher lengths of this GT repeat. When you look at the allelic differences, you can see that actually the American and African had the highest levels of the long repeats. Oh, let me comment on that. Typically around 25, 27 nucleotides is considered a dichotomous cutoff point, where shorter is considered short, longer is considered long. And so the data is represented based on that dichotomy characterization. And what you can see is that the long, which would have lower HO1 expression levels, is much higher in African and American/Hispanic backgrounds. And when you do pairwise comparisons between the groups, how we've represented that data here is if there was significant difference between the two groups, it's listed in red. Non-significant is in S, and the direction of the significance is by the larger font size. So in this situation, the short allele or the higher expression HO1 is higher in East Asians compared to Africa. So, and then when you look actually at the genotype per individual, that's represented in the panels below, looking at SS, which would have the highest levels of HO1 expression compared to the other genotypes, and LL, which have the lowest level of expression. And you can see that the distribution in the African population is significantly different than Caucasian or Asian populations. And also the Americas also is showing significant differences across the ethnicities. When we looked at the AT-SNP genotype, again, we saw differences among the racial ethnic groups. And here it isn't as quite uniform in terms of the outcome. Here, again, we see Africa having the longer or lower HO1 expression genotype. The American actually this time had lower compared to Asian and non-significant from European, and the Asian populations were both different compared to the European population. And what I also like to do is look at the combined genotype. So these are all the possible variations. You can see that actually in the human population, the SSAA genotype is likely lethal because it's not present within any of the human populations. SSAT is low frequency, but higher in Asian and European. Again, these are very small numbers in these cells, but what you can see is that there's clearly variance between the genotypes across the racial groups. So the question was the differences that are seen in ethnicity related to preeclampsia or other hypertensive disorders of pregnancy, could those account or associate with these hemoxygenase 1 genotypes? So we turned to the Preeclampsia Foundation registry, which had genomic DNA samples from women in the registry. And also associated with that was a lot of very granular clinical details about those pregnancies. And when we looked at the population cohort that we had to work with, what you can see right off the bat is it was not very diverse. So we weren't able to immediately look at whether the hypertensive disease in pregnancy correlated with HO1 related to race. But it did have a significant population of self-reported white that was also then confirmed by haplotype of the genomic DNA. So what we...
Virginia?
Yes.
Before you go to those results, Juno had a question in the chat. She was hoping you could speak to how these racial and ethnic groups were formed in the prior population. Yep. Are these really geographic groups or racial groups? Is American a stand-in for Caucasian? And what about African American in America?
Yeah, so the my understanding of the 1,000 Genome Project, and if Tien's on, she can speak up, is they included people who could trace their great grandparents on both sides to that region. And then I think they actually have validated a lot of it on haplotype. But it is confusing because the American is Central America and Southern America, so Hispanic. It is the European is the Caucasian, and then the African is a combination of Africa in the US and Africa in the African continent. Hopefully that answers your question, Juno.
Thank you so much, appreciate it.
Yep. So when we looked at, this is the frequency of the GT repeat of the hemoxygenase gene. And again, you see this similar histogram distribution. And in the AT, this is the frequency of the SNP alleles within this cohort. And if you look at the short long, there is a difference between the Preeclampsia Foundation cohort, and we compared this to just the European, so the white, the Caucasian sample from the 1,000 Genome Project. Obviously, there's caveats in terms of we don't have clinical data, so we don't know 100% whether they had a pregnancy, whether there was a preeclampsia pregnancy, but in terms of just a comparison to a general Caucasian population, you can see there is a difference in representation of the short and long alleles, not so much of a difference in the AT SNP. And when you look at the genotypes, you start to get into smaller numbers, so aren't statistically significant, but are trending to differences. And then I also, again, think it's informative to look at the combined genotypes. And again, you can see that there was no SSAAs at all in either cohort. And clearly the preeclampsia had higher numbers when it came to the long GT repeats. So this exploration into the hemoxygenase 1 genotypes showed that there is a heterogeneous distribution across racial groups, that the more protective SS-GT repeat is lower in African population and higher in the East and South Asian populations. AA was higher in the Americas and lower in African, and the LLTT, which would have the lowest level of expression of HO1, was highest in the African populations. You know, so does this genotype distribution have any explanation into racial differences? We're working on getting a second cohort of known normotensive pregnancy Caucasian women to do a further comparison with the hypertensive disease in pregnancy cohort that we had from the Preeclampsia Foundation. But this is all just clearly associations. And what we really want to know is, does the HO1 genotype really influence or impact a woman's risk of preeclampsia? And so that leads into our second project, which is basic science based, where we are developing a in vitro model system to really look at endothelial dysfunction in preeclampsia, and having this in vitro model be able to begin to dissect out both maternal contribution and genetics being brought to the pregnancy, as well as response of patients' endothelial cells to circulating factors and placental factors associated with preeclampsia. So the basis of this in vitro model system is that you can generate induced pluripotent stem cells from individual patients, either from blood or from cheek swabs. And recently, it's been determined how to culture these cells to get them to differentiate into endothelial cells. Because you can imagine it's really hard to study primary endothelial cells from a woman, because we're not really biopsying anything that can give us those cells. And then this becomes a renewable source for that individuals to generate endothelial cells to be able to do functional studies, so that we can really begin to dissect out endothelial dysfunction of preeclampsia. So this is just the protocol used to differentiate into the endothelial cells from the iPSC cells. And what we did for kind of a proof of concept and initial preliminary experiment was we took a female iPS cell line generated by our collaborator, Najish Azid, who figured out and developed the protocols for differentiating these into endothelial cells. And what he did was take these endothelial cells and culture them in the presence of normotensive pregnancy serum or preeclampsia pregnancy serum. And what you can see here, CD31 is a marker of endothelial cells that the preeclamptic sera downregulated CD31 to a much more greater extent than normotensive and compared to the control lines. CD144 and eNOS are also markers of endothelial cells, and eNOS is a marker of endothelial function. And you can see the same pattern by all of these parameters. And the serum, though, is not impacting the health of the cells in terms of number of cells. So this loss is not because of the sera killing off the endothelial cells. It is a reflection of these endothelial markers. And then more importantly, looking at endothelial cell function, a very standard assay is a tube formation assay, and that is shown here. If you plate these endothelial cells on Matrigel, they start to make this network. In the presence of control serum, you can see the network is not as robust, and with preeclampsia is clearly impacting these cells' ability to form these networks. And then this is just the quantitative data of those results. So based on this, we actually applied for a number of grants and were able to obtain funding both from the Bill and Melinda Gates and the Preeclampsia Foundation to build upon this in vitro model system. And so one of the aims of the project is really to isolate and generate IPS cells from women with normotensive pregnancies compared to preeclampsia pregnancies, differentiate these into the endothelial cells, and then carry out functional analyses. And we're interested in looking at the HO1 status. So we will genotype these cell lines and look at the expression of HO1 and both at the message level as well as the enzymatic level, and see how the genotype correlates in these endothelial cells with HO1 activity and the endothelial functional status. We're also then, so that's looking at the endothelial cells that the woman brings to the pregnancy. Then we'll also look at the response of these endothelial cells to sera from both control and pregnancy sera. And we will treat with a pooled sera, so that we'll have the same sera treating a panel of these endothelial cells from the woman to see the response, and again, look at both the HO1 pathway as well as a standard panel of endothelial functional assays. But then most importantly, really to assess whether these hemoxygenase variants are directly contributing to the endothelial dysfunction. We will be utilizing a CRISPR-Cas9 system to actually take one of the IPS lines, generate the different genetic variants within that same genetic background, and then repeat the studies that we would be doing on the individual cell lines and in the presence of the sera. So this will provide more definitive evidence of whether these HO1 variants really impact endothelial function and whether they show differential response to pregnancy sera and preeclampsia sera. So, oh, I've lost my ability to move. Hold on. Okay, so Tian has actually generated, oh, I missed a slide, has actually made good progress on this project. We have now 4 control lines, 4 preeclamptic lines. IPS cells have been generated. They have to actually be passaged to passage 15 before you can start to differentiate them into endothelial cells. They've all now been differentiated into endothelial cells in sufficient quantities for us to be able to move forward. And they've been fully genotyped in terms of these HO1 status. This is just confirming and validating her differentiation worked for each of the paired lines. The IPS cells do not express the endothelial markers, and the endothelial cells from that IPS line are showing endothelial markers. So the next steps really, we're just poised now to begin to look at, are there differences in endothelial cells from controls and preeclamptic women, whether the HO1 expression is different and how that changes in response to sera treatments. And we'll need to be generating those isogenic lines this year. So that's the exciting work that's happening on that project in the lab. Tian Sun is the postdoc who has done the work that I presented today. And I have a new postdoc that's just recently joined the lab, Yanming Wu, who will be taking over this project. And so hopefully we'll have an interesting report maybe next year to present. So I'd be happy to take any questions. And how are we doing on time? Oh, I did well. Great. Oh, I can see the chat now. Can I get to the chat?
Thank you so much, Virginia. I had one question. So if you're thinking that this the HO1 deficiency is kind of one of the things leading to preeclampsia, what are your thoughts about like potential treatment options? Like, is it a gene therapy, or is it something that affects the HO1, or will that lead to potential treatments?
Mm-hmm. So actually, that was one of the reasons I was drawn, besides Dr. Stevenson has examined and looked at this extensively and has a lot of expertise and able to do all of the HO1 assays. It turns out that aspirin and statins are known to upregulate HO1. And as we know, aspirin is really our only preventative therapeutic right now. And there's ongoing trials looking at the use of statins for preeclampsia prevention as well. So it's really wondering why those therapies may work is through their upregulation of HO1, and kind of the next step is going to be utilizing our in vitro model system to test how these drugs may be impacting the HO1 system. Could it be if you have a certain genotype, you're more responsive to aspirin or less responsive to aspirin? And is that explaining why aspirin doesn't help everybody in terms of preeclampsia prevention? So that's kind of the direction we're going. So there are drugs that are known to upregulate HO1 that are already being considered for preeclampsia prevention.
Virginia, hi, it's Ron. Very nice work to see this. I was wondering if you've investigated women with the clinical syndrome of gestational hypertension, and if so, is their pattern similar to women with preeclampsia or different?
Yep. So no, we have not, unfortunately, I actually have, I pulled it out, but let me just quickly show you, maybe not. Yes. So within the Preeclampsia Foundation registry, unfortunately, only 3 of the women had gestational hypertension. So we only have the genotype on 3. So you can see that, you know, this registry is clearly biased. It makes sense that people that had the most impactful and severe disease would be more motivated to register with the Preeclampsia Foundation. So what we're doing so we did a real quick look just between HELP and severe preeclampsia, since those were the numbers that could give us any consideration. You can see that there's not any real significant difference between those 2 in terms of their HO1 genotype. What we are also looking at is all the clinical granular data that we have with the Preeclampsia Foundation cohort. Are there any aspects of presentation of the preeclampsia based on genotype?
Yeah, it'd be nice.
Yeah, but I'm really excited to see how robust this in vitro model system can be, because we can look at not only HO1, you know, that's just kind of the first pathway to consider, but are there other pathways, and the fact that we can genetically manipulate the cells, we can do more definitive proof of how a woman's genotype impacts her endothelial health in response to pregnancy and normotensive versus preeclampsia. And then the other side of the fence, you know, we can begin to look at this the preeclamptic sera and begin to remove components to see if that accounts for the endothelial dysfunction, add in components, and really begin to dissect out on the placental side, what's contributing to endothelial dysfunction. I mean, we already know that the angiogenic imbalance factors should impact that, but when you look at preeclampsia, not all women that develop preeclampsia have elevated sFlt1 or low PLGF. So there's other pathways. And so we really need to get down to the individual level to begin to understand what these different potential pathways are, and how much is maternal contribution, which is versus how much is placental dysfunction contribution.
Virginia, Maury here. Thank you for such a great presentation. I just make a couple of quick observations that the carbon monoxide story is very interesting, because actually that paper came out of Stanford, and we were surprised when we saw that the CO was actually lower, because we thought it was going to be higher. And it probably accounts for the fact that people who smoke cigarettes may have a lower rate of preeclampsia. So that's interesting, because it sort of follows on the whole hemoxygenase pathway. And my question was on the nitric oxide. Is HO involved in generation of nitric oxide? I thought I saw that in your little thing there, but I wasn't sure. Is that correct?
No, HO is not. Where the nitric oxide was looking at the endothelium. So the endothelium with the endothelial nitric oxide synthase. So HO1 may impact that. I mean, that'll be one thing that we can look at. Does HO1 genotype impact levels of eNOS?
Yeah.
And therefore, you know, nitric oxide.
Yeah, the whole issue is that, as you said, you know, we keep looking at one piece of the elephant, the tail, the trunk, and it's hard to put it all together, because We did a study here many years ago where we gave patients with preeclampsia L-arginine, thinking that we would increase their nitric oxide and make the clinical phenotype better. It obviously didn't work because for many reasons, but it's so there's these simplistic approaches where you say NO is down, give them something to make the NO go up, or whatever intervention you do always fails because of the sort of multifactorial nature of this. So I think you just demonstrated that so beautifully with your work. So thank you. And the statin work is very interesting because David's lab, with Avitura showed in animals at least that the statins really work. And I think there's an NIH trial going on right now about statins and preeclampsia.
Yeah, there's actually a lot of beautiful work in a number of preeclampsia animal models suggesting that it's preventative. And there's both a UK study and an American study, both currently enrolling. One of them should be ending soon, looking at statin in pregnancy.
Great, thank you so much. Wonderful talk.
Thank you, Maury.
Virginia, I had a question for you.
Yes.
It's very nice presentation and quite a lot of progress in both areas of the talk. And my question was related to the hemoxygenase regulation. And I just wondered if you or anyone else in the literature had looked at the upstream transcription factor regulation, the NRF2, the AP1, and the hypoxia-inducible factor roles, and whether they varied in relationship to the prevalence of preeclampsia.
We have not, and I'm not aware of any literature, but there may be something out there, and definitely that will be something that we can look at using this model system.
Certainly, I'm not sure I would prioritize that hypothesis above yours because of the very suggestive findings you have on the SNPs and the GT repeats in relationship to the prevalence in the African American and African populations.
Yep.
Which is quite compelling. Lovely.
Yeah. Any other comments in the? Oh, I get to it this time.
Virginia, I have something. I'm like a total genetic novice. Like I don't.
Yeah, I'm not a geneticist either. This has really stretched my my expertise.
So I don't know if these are, it's just such naive or off the wall questions, but I'm wondering about kind of hormone level variation. It strikes me, I'm often asked in a very early pregnancy, right?
Yeah.
As with my gynecologist hat on,
about,
oh, should we do progestin levels? Should we do estrogen levels? For folks who are very early or have had pregnancy losses in the past. And we say, oh, there's such wide variation, it's really not helpful. We don't have good indices, but it's always bothered me that we should be able to actually sort of narrow in more. And I wonder about that with respect to, especially the link that you're talking about of what is set up very early in terms of vascular pathways and implantation and downstream effects to these later phenotypes, right? And so I'm just wondering about kind of some of those almost environmental or systemic, like, or, you know, the environment that, you know, or even things like methylation factors on these genes, like what, you know, or epigenetic kind of factors that are,
Yep.
you know, you could have that expression or not, but, you know, how those pieces, that's a very broad question, but it's just something that keeps going off for me in terms of shaping how this might come to pass in a human body, or like, you know, bigger system.
So actually, a more translational project. So we've also looked at endothelial, you know, one of our other projects is actually trying to assess endothelial function by a non-invasive method, and looking at the proteome, lipidome, endothelial and placental transcriptome in the EPUC study related to preeclampsia and then later cardiovascular disease risk. That doesn't directly answer your question, but we've been interested in that, and that interest came from a project looking at pregnancy outcomes in infertility patients that Val Baker did here before she went to Hopkins. And what we know is that IVF pregnancies have this, you know, twofold higher risk of preeclampsia.
Right.
But based on the observational data from that and some of the translational work we did on a sub cohort of that group, what became clear was it's actually not IVF, but it's suggestive that it's actually, when you do the transfer, are you doing it in a natural cycle or are you doing it in a medicated cycle? I.e., is there a corpus luteum or is there not a corpus luteum? And that actually that early pregnancy environment, which is what you were alluding to, you know, that early pregnancy environment is impacting, could that be impacting placental development or vascular adaptation to pregnancy? And we actually saw by the end of the first trimester differences in blood pressure related to whether there was a corpus luteum or not.
Wow. Okay.
And so that now has parlayed, which I'm super excited. I mean, it's so exciting for me as a basic scientist to actually see projects that I've done turn into randomized controlled trials that could change care. So Ruth Lochte is running the NATPRO study, which is now trying to prove that observational data, that it really is the environment in which you do the transfer. So they're actively enrolling. It's a 6-site across the country, actively enrolling here at Stanford for women who are undergoing a frozen embryo transfer. They're getting randomized to a natural cycle versus a medicated cycle.
Cool.
And we're going to be looking at preeclampsia outcome. What I should have said from the observational study, in a natural cycle, the rate of preeclampsia was no different than spontaneous pregnancy. In the medicated cycle, the rate of preeclampsia was 12%.
Wow.
Versus 4%. So, you know, that's observational data. You know, there could be biases on who got what kind of cycle. So that's what this randomized controlled trial will really be able to tell us. And if it is that big of a difference in preeclampsia risk in a medicated cycle, then immediately you can change care for anybody who can do it in a natural cycle. You'll take their risk of preeclampsia basically back down to the, you know, normal
baseline, or whatever.
Yeah.
Awesome. Thank you. That's really interesting.
And then, of course, I'm interested in all of the biologic things that we're collecting so that we can look at what are those corpus luteum factors that are different. You know, we know some of them, but we actually don't know as much about what the corpus luteum makes from humans as you would think, and whether those are impacting placental development. Yeah.
Thank you so much. Great.
All right, I think we're running out of time.
Great.
That's so inspiring. Thank you so much, Dr. Winn.
Yeah, thank you all for listening. Thank you for the great questions.
Bye.
Um,
Thank you all for joining.
And have a
See you
good
in December.
afternoon.
Thank you. Bye.